Hi, my name is Megan Kaplan, and today I will be giving a presentation about mapping the cancer genome. First, we must talk about the Human Genome Project, which gives us the basis for mapping the more specific cancer genome. The genetic basis of cancer was first found in 1981. Previously, they thought cancer was related to viruses only. The International Research Project to map the human genome started in 1984. It is important to note that the project was international because in the scientific community, we also often have a problem where scientists don't communicate as well as they should, stunting progress. However, this project was different from others where the scientists communicated all across the world. The main goal of this project was to map the sequence of all the nucleotide bases in human DNA. And the applied goal is to use the genetic information to find out what genes are responsible for cancer, a disease that has affected many people all over the world. Before I get into more specifics of how the Human Genome Project works, I'm going to clarify what the human genome is and some other terms that are related to genetics. The genome is defined as the complete set of genes or genetic material in a given organism. This image shows genetic material at various different levels. We have a strand of DNA where you can see the nucleotides it is made up of. A given set of nucleotide bases makes up one gene, and a gene codes for specific proteins. When we're talking about cancer, a mutation is going to affect an individual gene. In addition, we have chromosomes, which carry multiple genes. There are multiple steps that must be taken before the genes involved in cancer can be identified. First, we must collect large numbers of DNA samples. However, there's often confidentiality issues with doing this. DNA is the most personal part of a human being, it quite literally makes someone who they are. So people are often hesitant to give away their DNA, or they don't want to find out what all their genes are because it's such a personal decision. Next, we have to sequence all the nucleotide bases. There's multiple methods of sequencing the bases that I will talk about further in another slide. The sequencing was completed in 2003. By 2001, when they had only completed 90% of the genome, they'd already found 3 billion base pairs. However, they only found 30,000 genes, which is actually less than they had originally expected. Next, they must categorize the genes by function. And lastly, how this whole thing relates to cancer is they need to determine each gene's role in cancer. Once they've done that, they're going to create the Cancer Genome Atlas. And that would be really amazing because it would be accessible to people all over the world and really help further cancer research. The picture on the right of the screen shows the DNA double helix with its 4 base pairs: thymine, adenine, guanine, and cytosine. Now we're going to talk about the methods to mapping the human genome. So the 3 methods that were used in the 1980s and up until 2003 were linkage maps, maps of gene locations on specific chromosomes, and just sequencing nucleotides. So the picture at the bottom of the screen on your left shows a linkage map. And a linkage map demonstrates how to track a gene throughout a family history because genes are often heritable and cancer genes are particularly known to be heritable. And if you have one mutation on the gene already, people are more susceptible to get cancer. So the next one is maps of gene locations on specific chromosomes. And we know that all the chromosomes in our body are numbered. We get one from the mother and one from the father to make up our 46 chromosomes. And scientists have the ability to stain chromosomes and see each gene that is on each one. In addition, on the bottom right of the screen, we have an example of DNA sequencing, which is just the basics of how we know the order of base pairs in the human genome. Sequencing techniques have come a long way since scientists started sequencing the genome in 1984. They've become much more cost-effective and time-efficient, which was really helpful to the project in general because funding scientific research is difficult. So we discussed all about sequencing genes, but now we need to understand genes' role in cancer. So I'm going to give a brief overview of what cancer is and how people get cancer. So mutations occur often in the cells, however, not all of the cells turn cancers. Usually a person needs more than one mutation to have cancer, so there's the two-hit hypothesis, unless they're already genetically predisposed to it. For example, they're born with one hit or one mutation to their gene, and then they only need one more hit to get cancer. Mutations can be caused by a variety of things, including carcinogens, so being exposed to radiation or toxins, or errors in DNA processing. For example, when the DNA isn't repaired properly, or when DNA is copied and there's a mistake in that copying process. When a gene has been mutated, it becomes an oncogene, which promotes rapid, uncontrollable cell growth, and all of those extra cells end up creating a tumor. Usually the cell has regulated growth, and cells grow through a normal cell cycle, which involves apoptosis, which is cell death. However, in cells that are cancerous, they no longer go through a normal cell cycle. In addition, mutations can affect tumor suppressor genes, which are the genes that tell the cells to stop dividing. So we have cancer affecting the genes in multiple ways. It can increase cell division and take away the cues that tell the cell to stop dividing. So how can the Human Genome Project really make a difference in cancer? So we're going to think about how it can affect cancer therapies and help us find a cure for cancer, because that's obviously the end goal we all strive to achieve. So it gives us the ability to compare normal genes with cancerous genes. So we're able to take two samples of DNA, one from a normal cell and one from a cancerous cell, and analyze the differences between the two. Next, it's going to allow us to pinpoint which genes are involved with which type of cancer, because there's different genes that are going to be mutated that are going to cause different types of cancer, so we need to know those specifics. In addition, we earlier talked about how some people have a predisposition to cancer, and we would be able to screen them and see if they did have that predisposition to cancer sooner rather than later, because we would know what specific genes we would need to be testing for mutations. In general, for the public, we would be able to make tests that detect cancer, so we could detect cancer at earlier stages, hypothetically, and then make more effective treatments, because we know that cancer detected earlier usually has a higher success rate. Lastly, we could create a comprehensive list of genes affected by cancer, that project that we mentioned earlier, the Cancer Genome Atlas. And overall, we still have many questions about cancer, how different types of cancer work, and knowing the genetic basis of cancer and the specific genes that are involved with it would give us just a much better understanding of the disease itself, and can lead to many new therapies that would be very effective for many people. However, there are still some obstacles facing the project of mapping the cancer genome. First, it's hard to decipher which mutations are causing cancer or which mutations were the actual original cause of it. So once a cell becomes cancerous, there's going to be a lot more mutations after that. So pinpointing the original mutation on the given gene is going to be a difficult process. In addition, in one type of cancer, there can be a lot of different possible combinations of mutations. So we talked earlier about the two-hit hypothesis, meaning that there's more than one mutation needed for a cell to become cancerous. So it gets even more confusing to look at all the different combinations of mutations and which ones are actually going to cause cancer and which ones are not going to cause cancer. And next, we have many different types of cancer with many different types of mutations, and as we talked about, different combinations of mutations, which makes it almost go to an exponential scale of how many different mutations we need to look at. Because if there's specific mutations for each type of cancer, but there's also multiple combinations of mutations in a given cancer that will lead to the person being diagnosed with that cancer, we have a lot more mutations to consider to actually decipher which one is the cause of cancer. Cancer is individualized. There are many different types of cancer, but no universal cancer gene. Some cancers are extremely rare, which makes the treatment have to be even more individualized for that person because the incidence of that type of cancer is so low. In addition, despite if someone does have the same type of cancer as someone else, the disease itself can have an individual nature. We talked earlier about how there are multiple combinations of mutations that can cause certain types of cancer. So each person could have a different type of combination even though they have the same type of cancer. In addition, we often see different treatments affect people differently. From a broader view, everyone's physiological makeup differs, especially if they have any other pre-existing conditions besides cancer that affect the makeup of their body. So we must view cancer from the level of the individual. Also, at a cellular level, people's cells look differently. Some people have more malignant cells and some non-malignant cells, and especially when cancer begins, it starts to create kind of a chaos with all of the cells. So the researcher must be careful to look and decipher the cells in each individual very carefully. Lastly, cancer progresses differently in different people. We've all heard stories of people who have a prognosis of 1 year and they end up living 4 years. So we never know exactly the route of that cancer is going to take in that person's body. So we must take that into consideration when trying to just generalize cancer for everyone and instead take the patient for who they are and consider them. So for the last part of this presentation, we're going to talk about some of the structural barriers that affect our ability to do cancer research. So we're going to talk about it in terms of government and the economy. So the main points are that genetic testing and ethics and ethical questions it poses, legislation necessary for genetic testing, how to appropriately disperse the data that researchers come up with, and the funding of future research. So first we're going to talk about genetics and medical ethics. So there's always this question of how much knowledge is too much knowledge and how much we can interfere with our genes. So once we've found the genes that are involved with cancer, there's going to come up with multiple more questions of how much we should be interfering with those genes. Should we, if we see a baby in a womb with, let's say, one hit, because we will be able to do tests that tell us about the mutations of our genes of babies in utero. Should we be already interfering with their genetic makeup? And I think just knowing more and more about genes will pose more questions about medical ethics that often go into the realm of government and legislation. Next, we also have implications for public health and healthcare. So once we know the genes, we're going to be able to screen, hopefully, for all of the genes. And so should the government have to fund screening for all cancer genes we are looking for? How often should we have those screenings? So I think this is going to pose a new question and a new economic burden, possibly, on public health care. In addition, we need to remember that the information we have about the samples of DNA we're receiving is very personal to the people who gave us those DNA samples. So we need to appropriately disperse the data, which means controlling who has access to all the information we have about genetics. Should it be accessible to the whole general public or only medical professionals? And just being careful to make sure that everyone's personal information stays completely confidential. And lastly, we are on the tip of finding a cure for cancer based on so far what we know about mapping the cancer genome, but we still do need further research. So we do need government funds to support that further research. And as you can see in this picture, cancer should be an election issue, is what many people think. So we really need to find ways of getting the government's attention more about cancer research and how vital it is to finding a cure and how it really needs to be a priority in our lives right now. So in conclusion, the project of mapping the cancer genome gives us a lot of hope for the future of cancer and finding a cure for it. So I hope you enjoyed this presentation and learned a lot. Thank you.